Assalamualaikum and very good day. I am Mohamad Anuar from Center for Drug Research, University Science Malaysia Penang. I would like to share my research finding entitled Clitoria ternatea fraction ameliorated the hippocampal synaptic plasticity in the chronic cerebral hypoperfusion rat model, and this study is supervised by Associate Professor Dr. Zurina Hassan. As for the introduction, cerebral hypoperfusion happens due to the blockage in the internal carotid arteries and significantly cause reduction in cerebral blood flow, reduced oxygen internal supply, as well as ATP production. And this situation can cause progressive neural loss and the pathophysiological changes. We can see the accumulations of amyloid beta plaque, phosphorylation of the protein, white methylation, and also hippocampal damage. Hippocampal is crucial for learning and memory and also vulnerable to damage. So damage of this region can cause cognitive impairment that frequently observed in the dementia patient. So what is the challenges of dementia? We can see from the data from the WHO stated that 46.8 million people having dementia around 2015, and the number expected to be 74.7 million by 2030, and this number kept increasing until 131.5 million by 2050 people living with dementia around the world. So is there any cure for dementia nowadays? So what is our action? So in this study, we are providing preclinical investigation using nature-based product, which is Clitoria ternatea routes to reverse the cognitive decline in the chronic cerebral hypoperfusion rat model that may be beneficial for the future direction. And moving to the next slide, this is the plant of Clitoria ternatea, commonly known as butterfly pea. In Malaysia, it is known as bunga telang. It has been used for centuries as a memory enhancer, antidepressant, anticonvulsant, and sedative agent, and traditionally used in Ayurvedic medicine, antibacterial, antifungal, antidiarrheal, anti-inflammatory, and etc. It has a lot of phytochemical constituents such as clitorolactin A, cerebral loopule, stigmasterol, and cytosterol. And this is the objective of the study. First, we evaluate the effects of bioactive fraction from Clitoria ternatea root extract on in vivo hippocampal synaptic plasticity in the rat model of 2VO induced CCH. And objective number 2 is to measure the expression of the molecular synaptic signaling proteins following the treatment of Clitoria ternatea bioactive fraction in the hippocampus of 2VO rats induced CCH. And moving forward to the methods, and this is the flow chart how we conduct experiment. First, we develop bioactive fraction from Clitoria ternatea root extract, and this plant material was extracted using chloroform extract, and the phytochemical constituent in this extract was further separated using normal flash column chromatography, and we afford to produce 7 fractions, and we selected fraction 5 and 6 and combined them together to test on the animal study. And prior to animal study, we established the chronic cerebral hypoperfusion by 2VO surgery. And after 2 weeks of recovery period, we subjected the rats to the in vivo hippocampal synaptic plasticity by stimulating at the CA3 region and record at the CA1 region of hippocampus. And at the end of the experiment, we harvested the brain to study on the targeted proteins such as ERK, CRAB, BDNF, centophysin, PCN95, and CAMK2 alpha using Western blot analysis. Moving on to the result and discussion. So demonstrated the effects of CT root extract bioactive fraction of CTRF on hippocampal synaptic plasticity, as you can see from figure 1a. 2VO untreated rats suppressed in the input-output functions as compared to sham rats, and significantly reversed by single administration of CTRF 10, 20, and 40 mg per kg. And throughout the 2 hours of recording, 2VO untreated rats significantly impaired the LTP compared to sham-treated vehicle rats. And when we challenged with the different concentration of 10, 20, and 40 mg per kg of CTRF, was significantly able to maintain the long-term potentiation. To extend our findings, molecular work was carried out to measure the expressions of the proteins that correlated to the synaptic plasticity, include PSC95, centiphysin, BDNF, CRAB, ERK, and CAMK2 alpha. From the result obtained showed that decreased level of PS75, cytophysin, BDNF, and CRAB, and significantly reversed by single administration of 40 mg per kg, except for ARK and CAMK2 alpha. Despite lower expression of CAMK2 alpha after receiving CTRF 40 mg per kg, but in the LTP, showed that able to maintain the long-term potentiation. And this phenomenon revealed that reduced number of CAMK2 alpha would be sufficient enough to maintain the basal transmission. And moving to the next slide is the molecular mechanism of LTP induction, comprises of many events such as covalent modification of persisting proteins, the activation of cellular programs for gene expression, and increased protein synthesis. And as for the conclusion, the present study provides evidence that 2BO-induced CCH caused hippocampal synthetic plasticity impairment. This complement with our in vivo electrophysiology findings shows significant impairment in LTP. Administration of biodiffraction from Clitoria roots suggests that this treatment alleviated CCH-induced cognitive decline by increasing the expression of synthetic plasticity-related proteins. Hence, CTRF has the potential to be developed as a smart drug for the treatment of dementia related to cerebral vascular diseases. And we look forward to study on the pharmacokinetic and pharmacodynamic studies as well as the safety evaluation. And before I end my presentation today, I would like to thank you to RUI for the financial support and also the supervisory team. And that's all from me. Thank you for your attention.